Thank you, and thank you for organizing this, Mary, and it's an amazing meeting to be at, actually, and I hope the first of many here in Boston about personality disorders. So I am tasked to talk about genetics, and for those of you who may be following the field of genetics in psychiatric disease, it's a tough path to hoe. There's all kinds of complexity. People have been studying 64,000 schizophrenics and come up with relatively few targets. The field of genetics is complicated and wide, and so I've also, so there are a couple of ways in which the way I've structured my talk will actually meet some of the criteria that Mary described as the goals of this study, of this organization, I guess. So one is that I want to talk about the work of a junior colleague and help promote her work, because Mercedes Perez is done essentially part of the work that I'm going to talk about, and I will point you to her posters at the appropriate moment. But the other thing is that I've taken on a role as directing medical student education, and so I feel myself increasingly challenged to think about what it is that the neurobiological work that we do in personality disorders means to patients and means to the people who see the patients in front of them. Because I think very often we can get caught up in the interestingness of our biological findings and not think clearly about how we are contributing to the well-being of the people who we study and who motivate our work. So I have chosen to talk to you about just a couple of paradigms in genetics and to show you how, in a quite conceptual way, they might inform what we do. There are many more, rather than taking you through a list of candidate gene association studies that are promising in personality disorders, or particularly borderline personality disorder, or even through the novel and interesting preliminary data from a genome-wide association study, which Mercedes Perez Rodriguez is as a poster on. Actually, I point you to that poster and to a very interesting poster on opiate genes, which I'm not going to cover today. I'm going to cover other models and talk about how conceptually we might use those data to help our patients. So I will start by describing that the work that I do involves a lot of collaborators. And I mentioned Mercedes, but our whole group at Mount Sinai, who are well represented here, I have to say, Marianne Goodman, Aaron Hazlett, Harold Koenigsberg, Larry Seaver, and a group at the NIH who do the genotyping work. So we've talked a bit about borderline personality disorder. I think John Gunderson has pointed out relatively recently that borderline personality disorder is a problem that is one fundamentally of social interactions. There is, of course, affective instability, but it appears that there's really the affective reactivity occurs in the context of social interactions, as was so sort of beautifully described in Jill Hooley's study on the way patients hear praise. So, and it's likely to be, there's undoubtedly this disorder, like all psychiatric disorders and many, many medical disorders, is a product of gene by environment interactions. And undoubtedly, we used to think a gene by environment, then we used to think many genes by environment, then we began to know, of course, all about epigenetic changes. So I'm not going to try to be overly simple in that way. I'm going to focus particularly on oxytocin because it's an obvious one to study when we're looking at social interaction. We know, I think many of you know that in mouse models and in the prairie vole and many other mammalian models, oxytocin is a hormone that is pro-social. It's a neuropeptide that is released in particular interactions. It's released during breastfeeding, it's released during sex, and it's part of social bonding, maternal-child bonding, and it's been implicated in aggression and attachment. There are all kinds of things, and in fact, there's amygdala, we're back to the amygdala, oxytocin administration in healthy individuals attenuates the response to betrayed trust. So oxytocin is seems to be a neuropeptide that is an obvious one to study in a disorder that has so pivotal as its center difficult social interactions. And this is a model from a paper by Barbara Stanley and Larry Seaver talking about various kinds of peptides. So I wouldn't want you to think that I'm going to say that all of borderline personality disorder is about oxytocin. Far from it. There are opiate abnormalities that I actually think are quite salient, and Mercedes has some data on that in her poster. And there's been work done on vasopressin and oxytocin. That area has been probably the most studied for these neuropeptides involved in borderline personality disorders. So why would I begin to study genes? I mean, there's one thing we know, and this is, this by now seems old hat to us, but when I was training, and I like to think that wasn't a long time ago, although it's starting to be quite a long time ago, it was considered quite surprising that borderline personality disorder was heritable. Well, in fact, it's shown to be quite heritable, with recent studies showing heritability scores as high as 0.65 to 0.79 for the disorder itself. And elements, dimensions of the disorder are also quite heritable. And so, if things are heritable, let's begin to look for the genes. I asked some very simple questions here. So, is there evidence for an oxytocin deficit that contributes to the interpersonal symptoms of borderline personality disorder? If there is, how would we detect oxytocin deficits? And if there is an oxytocin deficit in borderline personality disorder, might the administration of oxytocin attenuate interpersonal symptoms in BPD? And I guess a further question that I didn't write here is, might we be able to predict from genetic data whether there are particular individuals whether there are individual differences based on genes that would suggest that one person might benefit more than another from drug administration. So why genetics? If we want to know if there's an oxytocin deficit, why don't we just measure oxytocin? That would be a good thing to do. But the problem is peripheral oxytocin doesn't actually reliably reflect central oxytocin. Oxytocin in the living brain can't be measured. You can't just go in and suck out a brain tissue and look at oxytocin levels. Cerebrospinal fluid oxytocin is studyable and, however, has not been a great marker in other disorders. It's been studied in schizophrenia, in depression, in OCD, and Alzheimer's disease, and it doesn't differ. It has not been studied in borderline personality disorder, in part because I think the studies of oxytocin in CSF came from older studies. I think people are increasingly loath to be willing to have a spinal tap, essentially, for the purposes of research, and I can well understand that. So it has been shown cerebrospinal fluid oxytocin levels have been shown to correlate with suicidal intent, with a history of emotional abuse, and to correlate inversely with lifetime aggression. So some of the features of borderline personality disorder have been associated with variants in oxytocin level centrally in the cerebrospinal fluid. Surprisingly, so what's another way of getting at it? If we can't actually measure it directly in the organ target we want, why don't we look at the genetics? So surprisingly, there have been no genetic studies examining oxytocin in borderline personality disorder to date. We know that there are two primary proteins that get studied when people look at genetics of oxytocin. One is the oxytocin receptor gene. That's the receptor on the cells that the oxytocin binds to and has its effect. The other is the gene that makes the oxytocin neuropeptide, the oxytocin gene itself. And so the oxytocin receptor gene has been shown to be associated with empathy, with aggressive behavior. There have been variants, and what this essentially means is that minor genetic variances in the oxytocin receptor has a behavioral stamp. A particular variance has been associated with prosocial temperament. So what I don't know how many of you know a lot of genetics. What we're talking about are simply in common variances. So the human genome is very complicated, but when you look at the oxytocin receptor, there's a set of genes there's a gene that encodes for it. And the way most genetic studies, candidate gene studies, are done is that there are little pieces of DNA that anneal to the native DNA and mark it, but you have little SNPs, little pieces of DNA that adhere to it, so we can pick up common variances. That doesn't mean that we pick up uncommon variances. It doesn't mean we know exactly what's happening in the gene, but that's what these are done. So this is a way of studying common variability in populations. In the oxytocin gene itself, there was a study looking at antisocial and aggressive behavior in children that did not find an association with variants in the oxytocin gene using SNP analysis, but another one that showed sort of a dopaminergic response to stress. So that's what little we know about oxytocin as genes as it relates to borderline personality disorder. So I thought I'd tell you what we have data on is actually the oxytocin gene in borderline patients. And so we don't actually have data on the receptor. We have data on the gene for oxytocin. It's a nonapeptide. It's mapped onto chromosome 20, and it is a small gene, actually, and highly conserved across species. So there's a very limited amount of inter-individual variability. So that means, and a little bit of maybe genetic lesson, I'll tell you about that. That means that it's a very small gene that's highly conserved. It means it's extremely important for our long-term, for the survivability of human species. It's not surprising if it's involved in breastfeeding and sex. It's not surprising that it would be highly conserved in human beings as well as all other mammals. But it's also easier to study in the sense that there are not that many SNPs, and it's a sort of limited gene to study. So we actually have these data from these 4 SNPs that came out of a study with David Goldman. These are SNPs that were selected because they'd been implicated in some sort of behavioral response. So we studied these 4 SNPs and in 156 healthy controls, 181 patients with borderline personality disorder, and 196 subjects with other personality disorders. Now, this sounds like a big group of people from the point of view of the kinds of studies we've been talking about today, but from the point of view of genetic studies, this is a really small group. And it becomes more complicated because genetic variants, so it's preliminary, the data I'm showing you. The genetic data becomes more complicated because you have to, in order to look for genetic variants that's meaningful, you actually have to look at genetic pools that are common, that share. So you don't want the ethnicity of the group to overwhelm the signal related to disease, essentially. So therefore, when you do a genetic analysis, particularly in genes that are variable between ethnic groups, you really need to have a sort of homogeneity in your ethnic background of your sample. So there are ways to do that. One way is to ask people what their ethnic background is. It turns out that Caucasians of European ancestry are relatively homogeneous. Turns out New Yorkers are very, very heterogeneous. So it makes your study sample smaller. When we say we are who we think we are, we're really not often. So we actually, we're all, I myself, and certainly my children are the product of the hybrid vigor of the American experiment. We have many, many different ancestral roots. So, but having said that, we therefore in New York have to use what we call ancestry informative markers to say, are we really who we think we are? And let's establish that we're Caucasian, have these European ancestry informative markers, so our sample became smaller. So what we did is we had some data, we had some, the relevant data that we had on the sample were related to social functioning, was the Bus-Perry aggression questionnaire, the Barrett impulsivity, and we had the experiences of close relationships inventory. So our question, and the experiences of close relationships inventory really tells you something about the attachment style of the individual, whether they had an avoidant attachment or an anxious attachment. And it's been pretty well validated. So what did we find? We started with a SNP analysis, which is simply looking at common variances related to these 4 SNPs that I described. They are related to impulsivity. We found a suggestive signal with one of the SNPs suggesting that the TT allele was somewhat related to an increase in BIS. That's a relatively weak finding. There's not a big effect size, but we did have some association with the BIS. The other association that I actually find more compelling is that in a particular SNP, this RS274210, we found a linear relationship between the presence of the G allele and aggressive scores. Now, another piece of basic background in genetics, we all have genes from our mother and genes from our father. So we all have, in any given SNP, we have TT and TG and GG. It means that if you're homozygous TT, it means you got the T from your mother and the T from your father, or you might be a mix, or you might have both G. So what you learned in sort of, I would guess by now, 6th grade genetics, I think that I have an 8th grade child, I think she learned this in 6th grade, is that, you know, genes are dominant or recessive. That turned out not to be true. Some genes are dominant, some genes are recessive, some genes are neither. And this would be evidence of a gene in which having one copy makes a difference and having 2 copies makes more of a difference. So it looks to me that the aggression is associated with the G allele, and having 2 copies of the G allele makes you more aggressive. So this is a suggestive finding that this kind of variance in the G allele may increase your risk for aggressive behavior. Now, this is actually data across the sample of European, of the homogeneous sample of people of European ancestry. This is not related to diagnosis. This is related to the dimension of aggression. We did not find an association between this SNP, between any of these 4 SNPs in borderline personality disorder. Now, in order to sort of try to give more power to a sample like this, we do something called a haplotype analysis. So what a haplotype analysis is simply taking genes. So I've told you that the oxytocin gene is conserved. What that means is that it doesn't change, it's conserved for hundreds or perhaps thousands of years. And the pieces of the gene that are conserved, where there hasn't been a lot of change. You all know that the gene changes because we have recombination, and recombination is how we all develop this hybrid vigor. But in fact, there are parts of the gene that are so important that we don't have that much change. And those chunks of DNA held together in important genes are called haplotypes. That's what a haplotype is. A haplotype are these set of DNA variations which are conserved relatively, where there's relatively limited variance. And so if you can find haplotypes and do a haplotype analysis, you can actually get a more powerful signal related to behavior. And so we did a haplotype analysis, and the advantage of the haplotype is if you find something significant, it's much more meaningful. The disadvantage is haplotype means you have chunks of genes running together, so there are lots of possibilities, and you only want to look at the ones that you have a substantial number of in your data set, and that means you have to throw out some of your data, because some people will have a very uncommon haplotype, and then you don't really know what to do with that, so those get excluded. So having been conservative about my description of this, we did in fact find that 2 haplotypes that are extremely interesting as they relate to aggression, actually, to the BPAQ scores. We found that there's this CT haplotype, and that means that either you have this haplotype, you either have it or you don't have it, or you have it on one gene and not the other, and so it's a mix. And the haplotype means that you have this chunk of DNA together, a chunk of genes being inherited as a group, and you have it either not at all on one copy or 2 copies. And we found a pretty robust association between the CT haplotype and aggression on the BPAQ. So those people with 2 copies of this CT haplotype were actually very much more aggressive, quite significantly more aggressive, and surviving correction for multiple comparisons. So this looks like a risk haplotype for aggression. We found a similar relationship with the ECRI, which for this same haplotype for attachment anxiety. So, and in fact, so this haplotype looks like a risk for aggression and a risk for anxiety. And so those are our data on this particular haplotype. And so that might be a risk allele in which, let's say, these people have, and we don't have clear measurements of the effect of the haplotype on peripheral oxytocin, but having that risk haplotype may indicate it's the kind of person who would respond to oxytocin administration. We don't know that. That would have to be tested. But that's the kind of thing, and I'll tell you a little bit about how you might begin to test oxytocin and what's been done, because it's a lot harder to do genetic analysis of this kind of thing than it is actually to just administer oxytocin, which is a relatively safe drug. Problem is oxytocin doesn't work in everybody. And so the reason we might do such a study is to say, okay, I've explained to you a little bit about how we do this analysis, and if we can find that there's a haplotype that puts people at risk for aggression and anxious attachment, that might be the group of people that we'd say, okay, these are the people that are going to respond more to oxytocin. And it might explain some of the complexity, which perhaps I should have described already, but I will describe to you. There have been studies of oxytocin in borderlines which go both ways in terms of whether people get better or worse, and I'll show you that data. But so then we found this risk haplotype. We also found a protective haplotype. So we found that the presence of the 2 copies of this CC haplotype actually conferred a greater benefit, lower BPAQ scores, and non-significantly lower. I mean, it's a little bit of a different path for the BIS. There was no relationship in this protective haplotype with anxious attachment. But at least in terms of aggression, this looks like a protective haplotype. This might be a group of patients. If you find a patient with borderline personality disorder with the risk haplotype and not the protective haplotype, then that this might indicate an individual who might not be the best candidate for oxytocin. So back to a simple question. Is there evidence for an oxytocin deficit that contributes to the interpersonal symptoms of BPD? Maybe, but no direct evidence since we haven't measured oxytocin and BPD. How would we detect it? Well, I've said to you that genetic haplotypes may be the way to identify people at risk for this particular deficit in oxytocin. And I've told you that there's a particular haplotype that might be a risk and one that might be protective. So that might give us a way of a biomarker for who might be at risk. So what about giving oxytocin to borderline patients? Oxytocin is actually a pretty easy drug to get. You actually take it. The way it's, recently in psychiatry, we've been doing intravenous ketamine infusions. Oxytocin is not such a difficult drug to take. You snort it, and it's not that expensive, and it's synthesized, and so not bad, okay? So people have done some studies with oxytocin. I confess to you that lots of oxytocin lies around my office. There are people using it next door to me, and a lot of my colleagues have tried it. I confess I have not tried it. But I hear, and it is studied, that it has, that in healthy individuals, intranasal oxytocin is anxiolytic. But it has been used in patients with borderline personality disorder. And the studies have shown different things. So some studies have shown that, that oxytocin is stress-reducing. Daphne Simeon showed when you administered oxytocin to borderline patients, they felt a subjective decrease in stress. It was anxiolytic. Sabina Herpert's has presented at meetings, but hasn't yet, that I've seen published, data showing that oxytocin in borderline patients actually attenuates the amygdala hyperactivity she finds in borderline patients, although you've heard from Marianne that not every borderline patient has amygdala hyperactivity. But Jennifer Bartz did a study in which she had borderline patients playing a trust game. So it's essentially a game theory model where you play with a counterpart and you have to you entrust money to the other person, and then the other person invests money back in you, and it's a game essentially of cooperation. And so the idea is, okay, oxytocin is pro-social, so maybe if you give people oxytocin, they're going to cooperate more, and when you cooperate more on this game, each party, by the way, earns more money. So we thought, okay, maybe that would work. It turns out for healthy people, you give them oxytocin and they got a little better. But patients with borderline personality disorder showed less cooperation and less trustfulness when they were administered oxytocin. Specifically, those with an anxious attachment style did worse on this game. So this, there's been some data on the schizophrenia spectrum which has been more consistently positive. And I'll show you this because I'm going to show you some data on schizotypal patients. But so what that means, I mean, when you look at this data, and perhaps I should have said it earlier, is so if you just give oxytocin to borderline patients, it's not obvious if they're going to get better or worse. And some people have shown, yeah, they get better. So there's a lot of reasons why that might be true. I mean, there might be a context issue. So the effects of oxytocin, I left off, of course, the most important center thought of my study. It might depend on diagnosis, right? You know, healthy people might get better, borderline patients might get worse, but maybe not. It depends on the context. So maybe in the context of trying to being pressed to cooperate, it's important. Oxytocin may make people worse. And in fact, when Jennifer came to me and said, she worked with us at that time, said, oh, I think I'm going to give oxytocin to borderline patients. And I said, actually, they make it worse because oxytocin draws you to other people. And I thought it might make patients more anxious and more intense in their interaction. In fact, that happened. It might depend on attachment style. It might depend on genotype. But I can tell you that part of the importance of studying genes and looking at that, genes as an individual difference, is that people are using oxytocin all over the place now in borderline personality disorder because it can be prescribed clinically. And we really have no idea whether it works. So it has to be studied, but I think it has to be studied with these other features, with the features including genetics potentially as a marker. So how might we study this better? So I'm now going to point you to a potential model for how you might study oxytocin in a way that gives us a more reliable signal. One thing I would tell you is get the genotype to see whether the genotype actually the haplotype has an effect. And the NIH would like us to select our patients based on haplotypes and that sort of thing, but I think we haven't found a way to do that. People come in complaining of symptoms. They don't come in telling us what their genotype is, but we are not going to do it that way, but we will genotype our patients. But I'm again going to highlight Mercedes' work because she's really planning a study in which we have have you all heard of the MASS? Because just let me have a sense. Do you know what the movie for assessment of social cognition is? It's been done, and I'll give you some data on where it's been done, but it is a more complex social cognition task. So this might be a way of developing a context for, so we've talked about diagnosis, context, interpersonal attachment style, and genotype as ways of determining when oxytocin might work. Well, we can potentially control at least the genotype and the context through this study. So this is a study in which patients watch a 15-minute movie and it goes along, it progresses, and then there are 45 questions. The movie stops. You have to answer the questions about what you think the character's feelings, thoughts, and intentions are as the movie progresses. And this is an example which may work, may not work. Let's see. Now, why does it work but when we're trying it at lunch? Any ideas? What I can do is we have it on another, I wasn't planning and hoping not to have to do this, but in fact we have it on another slide. Hold on, I'll get it for you. Okay. No, mask. Let's see. Do you know where you put it, Mercedes? No. It was embedded, but it was also elsewhere. We had it
under my documents.
Just under my docs? Okay.
So over here.
Oh, it's just under my docs rather. Okay.
So I guess if you order by date modified,
Yeah.
it should be
Right here.
one of you.
All right, we're trying
by
not,
name.
okay, there we go. All right. So we're going to show you the movie for social cognition, which somehow when we import it on this computer, possibly because I imported it from a Mac.
Hey, you. Surprise? I thought this was where you live.
So how are you?
I'm fine. But take a look at you. You look terrific. New hairstyle?
Yeah.
Classy.
Thank you.
Okay. So the question, okay, now let me get out of here. So that's a little snippet, okay? So, let's see. So the question, having now said that, is, okay, So we saw a little snippet, and the question is, what was Sandra feeling? A, she's flattered but somewhat taken by surprise. B, her hair doesn't look that nice. C, she's pleased by his compliment. Or D, she's exasperated about Michael coming on too strong. So these aren't so simple, right? These are not, I mean, there are many ways of thinking about this, and these are the way this is scored. The optimal answer is she's flattered but somewhat taken by surprise. That would be considered the optimal response as scored by this. Her hair doesn't really look that nice would be kind of the concrete response, the non-mentalized response, ignoring the social interaction. The hypomentalizing error, I'm reminded of this, and I shouldn't tell a story. I'm reminded that I once gave a talk at the ACMP with my mother in the audience. My mother is a very famous scientist. And I guess I may hypermentalize, because as I was walking up to the podium, she said to me, "Antonia, fix your hair." And I did not take that to be about my hair. So having said that, it wasn't about my hair. At least I don't have a non-mentalizing response. I might hypermentalize. Hypomentalizing, so she's pleased about his compliment. Maybe not, right? And hypermentalized is she's exasperated because he's coming on too strong. So these are a series of questions, and it's hard. Not everybody gets, I mean, healthy people don't get the optimal response, but it's a relatively difficult task that sets a context that gets people emotionally involved to some degree. So it really, you can get a measurement from this task of mentalizing accuracy, and borderline patients in previous studies, I don't know if I'm saying Priesler right, is that correct? But borderline patients are less accurate than healthy controls, as are people with schizophrenia, where it's also been studied. Individuals with schizophrenia make more hypomentalizing errors, whereas individuals with borderline personality disorder make hypermentalizing errors, as Dr. Sharp knows. So, and her data show. So the plan, and this is one of the ways that Mercedes is actually proposing to do, is to look at this in the context of using oxytocin to see whether oxytocin actually changes mentalization. And it's an open question. We have some preliminary data that she developed showing that in a small sample, really pilot data for a grant application, for borderline patients and 14 controls recruited actually through a VA study. And the controls are, by the way, psychiatric controls. These are all veterans. And preliminary data, so what we see is, as predicted, the accuracy of the patients with borderline personality disorder on the right, I guess I can, does this, let's see. So borderline patients with borderline personality disorder, veterans, are less accurate than non-borderline veterans, all of whom have significant psychiatric illness, by the way. So that's quite a robust difference for a psychiatric control sample that's actually equally sick. And we show that in fact, though this is not statistically significant because it's a very small sample so far, the individuals with borderline personality disorder make more hypermentalizing errors. I'm sorry that that Fiona Graff isn't here. She's coming. I guess her poster is here. So I'm going to direct you yet to another poster for one of our junior colleagues, which looked at actually, which she has data on schizotypal personality disorder, and actually individuals with schizotypal personality disorder don't differ from controls from healthy controls in their hypermentalizing, but in fact do hypomentalize like individuals with schizophrenia. So that the model for one way to look at this is, first of all, to say this would be the overall design of a study, which is that there's hypomentalization and hypermentalization, and that we hypothesize that borderline patients hypermentalize, schizotypal patients, schizophrenia patients, and people with autism spectrum disorder might hypomentalize, and healthy people would be in the middle. And the question is, when you give them oxytocin, because we're talking about oxytocin treatment, does it happen like this? Do these individuals who hypomentalize, when they get oxytocin, Do they optimize more optimally? And borderline patients who hypermentalize, do they also optimize their mentalization? We don't know. We need to know context. So it's probably context dependent. It may be genotype dependent. Or instead, does everybody mentalize more? In which case, borderline patients will fall off the curve and get worse. So, Dr. Bartz's data suggests this. Dr. Simeon's data suggests the former model. And we don't really know the answer to that, and that's why I'm very optimistic that Mercedes grant will be funded and we can find that difference. So this is I'm going to go through this rather quickly. So back to the simple question. So if there's an oxytocin deficit in borderline personality disorder, might administration with oxytocin attenuate the interpersonal symptoms? This isn't answered yet. This is why we need more research. But I gave you a sort of possible path of a model in which we can control context and activate people's social interactions with the mask, and in fact use oxytocin and potentially control for genotype and actually predict what happens. So that's data to be studied. Now, right, thank you in there, but there is, in fact, I think I don't have my most recent. Mary, I can either go on and tell you a second model, because somehow my second model is not here. I can, all right, hold on. Do you want me to go on and do I have a little more time, or because, no, okay. So, I will tell you without slides a second model, just because I think it's a promising other way to go about this. And that is simply that you heard about the habituation failure. I mean, always inevitably you can look at the relationship of genes and think about how do genes relate to the biological findings that we have. We have found there's a lot of interest. There's been 10 years of people interested in BDNF, brain-derived neurotrophic factor, as the disease du jour, and I mean the molecule du jour. And in fact, I've been hearing about the data on habituation we've been studying this for a couple of years, and Harold Konigsberg's data is actually an independent replication sample within our group showing a habituation failure in borderline personality disorder. So we think that's robust, but we knew little about the molecular underpinnings of this habituation. What is habituation? What controls that in people? Well, recent data showed that BDNF actually has an effect on habituation. So we said, oh, we have habituation data, we have BDNF data. So we actually looked at the BDNF genotypes in habituation, and we found a pretty robust effect for specific BDNF Snip and habituation. So we actually found that there was that BDNF genotype predicted habituation in our sample in Erin Hazlett's sample. So the reason I point to that is that's yet another opportunity to consider treatment targets. If that's true and habituation failure relates to DBT response, then potentially we can augment DBT response by enhancing BDNF. How do you enhance BDNF? There are very traditional ways, like with lithium, which we actually have tried already in patients with borderline personality disorder, antidepressants. Actually, olanzapine dramatically increases BDNF, and Mary's shown that olanzapine's effective. There are other things too, though. There's Q-Cucerin, which is a component of Indian curry, which enhances BDNF. And there is a component of Cabernet Sauvignon, which actually enhances BDNF dramatically. And so I'm not actually going to recommend that all of you send your patients who are in DBT home to have a bottle of Cabernet Sauvignon, because they'd actually have to have that much. But there is available as a sort of alternative therapies. These are ways of enhancing BDNF that might, in fact, be integrated into the treatment, into DBT treatment that are that are safe and can be given relatively easily. So this kind of genetics research can actually point us to novel approaches, even though right now there's no sort of novel BDNF, there's no novel drug that specifically targets BDNF, but it happens to be relatively easy to target with nutritional supplements. So having said that, I'm going to stop there and ask for questions.